friends, welcome back to my channel. In this video, I am going to give you a brief lecture on mechanism of aerobic respiration. So, the aerobic respiration involves four major pathways. They are, first one is glycolysis. So, this is the first step of aerobic respiration. Here, one glucose molecule splits into two pyruvic acids. Glucose is a 6-carbon compound, whereas pyruvic acid is a 3-carbon compound. This is the first step of aerobic respiration. So, this step occurs in the cytosol of the cell. So, in this step also, some amount of ATP is released through the substrate level of phosphorylation. Let's see the second step. In the second step is oxidative decarboxylation of pyruvic acid or pyruvate. So, second step is oxidative decarboxylation of pyruvate. In this step, the pyruvic acid is prepared in the cytosol that enters into the mitochondrial matrix. In the second step, the pyruvic acid undergoes oxidative decarboxylation. Okay, two pyruvic acids undergoes oxidative decarboxylation results in the formation of two acetyl coenzyme. So, which is a 2-carbon compound. So, this is the second step. There, two pyruvic acids undergoes oxidative decarboxylation resulting in the formation of two acetyl coenzyme molecules. So, this step occurs in mitochondrial matrix. So, let's see the third step. Third pathway is nothing but Krebs cycle. This is also occurs in mitochondrial matrix. So, two acetyl coenzyme A. So, they completely oxidized, resulting in the formation of CO2 and H2O. So, acetyl coenzyme A present in the mitochondrial matrix, they are completely oxidized through a sequential steps, that is nothing but Krebs cycle. So, there they completely oxidized to CO2 and H2O. Okay, during this process, energy molecules are released. They are nothing but NADH plus H plus FADH2. These are the energy molecules released in the TCA cycle or Krebs cycle. Now the fourth step occurs, that is electron transport chain. In the electron transport chain, so these energy molecules, when they passing electrons through the membrane, they releases energy. That energy will be utilized by the ADP for the synthesis of ATP. So, this kind of ATP preparation is called as oxidative phosphorylation. So, let's see the first step in detail. Let's see the glycolysis in detail. So, glycolysis is the first step of aerobic respiration. So, it occurs in the cytosol of the cell. So, here one glucose molecule splits into two pyruvic acid molecules. So, it's a Greek word actually. Glycolysis is a Greek word. Glykis means sugar. Lysis means splitting. So, glycolysis is nothing but splitting of sugar molecules. Here, one glucose can convert into two pyruvic acids. One glucose can break down into two pyruvic acids. Okay. So, it involves so many steps. Those steps were identified by three main scientists, three German scientists. They are Emden, Meyerhoff and Paranas. So that's why this pathway is also called as EMP pathway. Embden-Meyerhoff-Parnas pathway. It is also called as a Hexose Diphosphate pathway. Why? Because fructose 1,6-bisphosphate is a key intermediate product of glycolysis. That's why this pathway is also called as hexose diphosphate pathway. It occurs in both aerobes and anaerobes. So in the case of aerobes, if you see aerobes, glucose can convert into pyruvic acid. This enters into mitochondrial matrix. There it can convert into acetyl coenzyme A. So this condenses with oxaloacetic acid and it is completely oxidized to CO2 and H2O in mitochondrial matrix. In this process, energy molecules, highly energy molecules NADH2 and FADH2 are released. So they enters into electron transport chain. They releases energy that will be utilized for the synthesis of ATP. This kind of ATP preparation is called as oxidative phosphorylation. Okay, so this is called glycolysis, this is called Krebs cycle, and this is called electron transport chain. Okay, so now we are dealing the first step, that is glycolysis. So let's see the pathway in detail. Okay students, this is the glycolysis pathway. There the 6-carbon compound glucose can convert into 2-carbon compound, that is pyruvic acid. Totally it consists 10 sequential steps. So let's see the steps one by one. So let's see the first step. So there one glucose phosphorylated into glucose 6-phosphate. The first step, glucose is phosphorylated. That step is called as phosphorylation. There one ATP will be utilized. It converts into ADP. Okay, glucose converts into glucose 6-phosphate in presence of an enzyme hexokinase. So let's see the second step. Here glucose 6-phosphate can convert into fructose 6-phosphate in presence of an enzyme isomerase, because these two are the isomers. Isomers nothing but glucose consisting aldehyde group and fructose consisting keto group. That is only the difference in their structure. That's why both are isomers. In presence of an enzyme isomerase, glucose 6-phosphate converts into fructose 6-phosphate. So let's see the third step. This is also phosphorylation step. Here fructose 6-phosphate can convert into 1,6-bisphosphate. This is also phosphorylation step. Here also one ATP is utilized. So then fructose 6-phosphate converts into fructose 1,6-bisphosphate. This is the key intermediate product of glycolysis. That's why glycolysis is also called as hexose diphosphate pathway. Okay, the enzyme catalyzing this reaction is phosphofructokinase. Phosphofructokinase that catalyzing this reaction. So let's see the fourth step. Here fructose 1,6-bisphosphate that breaks down into 2 3-carbon compounds. They are nothing but GAP and DHAP. Glyceraldehyde phosphate and dihydroxyacetone phosphate. Actually these two are also isomers. Why? Because in dihydroxyacetone phosphate there is a keto group. Glyceraldehyde phosphate there is a aldehyde group. These two are the 3-carbon sugars. So in presence of an enzyme aldolase, fructose 1,6-bisphosphate that breaks down into 2 3-carbon compounds, nothing but GAP and DHAP. Okay, let's see the fifth step. So these two are the isomers. Isomers nothing but they can interconvert. Okay, so they are having only one functional group is the difference in their structure. They are only having the difference in their functional group only. In the fifth step, DHAP can convert into GAP in the next step. Okay. So from here to 10th step, from 6th to 10th step, the reactions occurs 2 times. Why? Because first one GAP undergoes the reactions. So then DHAP also converts into GAP. So that's why from here to 10th step, the reactions occurs 2 times. Okay, because DHAP also converts into GAP, again these steps occurs. Okay, so let's see the sixth step. So the sixth step is oxidation reaction. Here one NAD plus can converts into NADH2. Okay, so here one inorganic phosphate is added here. That's why glyceraldehyde 3-phosphate can converts into 1,3-bisphosphoglyceric acid. Okay, in presence of an enzyme dehydrogenase. Okay, in presence of an enzyme dehydrogenase. So let's see the seventh step. So seventh step is dephosphorylation step. Here you will get 2 ATPs actually. In presence of an enzyme phosphoglycerokinase. So 1,3-bisphosphoglyceric acid is converts into 3-phosphoglyceric acid. Okay, so this step is called as dephosphorylation step. So next step, 8th step, that is nothing but intramolecular shift. There 3-PZA can converts into 2-PZA. Okay, 3-PZA converts into 2-PZA. Nothing but the phosphate group shifted from third carbon to the second carbon. That's why 3-PZA converts into 2-PZA. So intramolecular shift in presence of an enzyme phosphoglyceromutase. It occurs in presence of an enzyme phosphoglyceromutase. Okay, so let's see the 9th step. Here one water molecule given out. So this step is called as dehydration step. 2-phosphoglyceric acid can converts into 2 molecules of phosphoenol pyruvic acid in presence of an enzyme enolase. In presence of an enzyme enolase. That is the 9th step. Let's see the 10th step. This is also dephosphorylation step. Here phosphoenol pyruvic acid undergoes dephosphorylation. Here also we are going to get 2 ATPs. Okay. So phosphoenol pyruvic acid converts into 2 molecules of pyruvic acid. So finally we are getting 2 molecules of pyruvic acid. So let's see the energetics of this cycle. So in the first and third steps, 2 ATPs are utilized. Okay. So in the 7th and 10th steps, we got 4 ATPs. Okay, here we get 2 ATPs and here also we get 2 ATPs. Totally 4 ATPs. So you have to subtract 2 ATPs from 4 ATPs because in the preparatory phase 2 ATPs are already utilized. That's why we have to remove 2 ATPs from 4 ATPs. That's why the net gain of ATP from glycolysis is nothing but 2 ATPs. We only get 2 ATPs from glycolysis. So we get energy molecules in the 6th step. That is nothing but NADH2. NADH plus H plus. Okay. So that the reaction occurs 2 times. That's why we get 2 NADPH. Okay. So 2 NADPH and 2 ATPs. That is the energetics of glycolysis. Okay. So let's see each step in detail. Okay. So, okay students, let's see the steps involved in the glycolysis. Let's see the 10 steps. So, in the first step, glucose undergoes phosphorylation. In this step, 1 ATP is utilized. Then, glucose converts into glucose 6-phosphate. In the second step, glucose 6-phosphate undergoes isomerization. There, it converts into fructose 6-phosphate. This step is called as isomerization. So, in the third step, fructose 6-phosphate undergoes phosphorylation again. There it converts into fructose 1,6-bisphosphate. So which is a key intermediate product of glycolysis. That's why this pathway is also called as hexose diphosphate shunt. Okay, so let's see the fourth step. There fructose 1,6-bisphosphate so is cleaved into, that's why this step is called as cleavage, is cleaved into 2 3-carbon compounds, that is GAP and DHAP. GAP and DHAP are also isomers. Why? Because the difference in their structure is functional group. GAP consisting aldehyde group, whereas DHAP is consisting keto group, C double bond O. CHO is the aldehyde group. So the fifth step is isomerization again. So there DHAP, which is a 3-carbon compound, it converts into GAP. In the next reaction, so only GAP is participated. So in the sixth reaction, GAP undergoes oxidation, oxidative phosphorylation. Okay, so one phosphate also added to the GAP, then GAP converts into 1,3-bis-PGA, bis-phosphoglyceric acid. This step is called as oxidation. In this step, one NAD converts into NADH plus H plus. Okay, let's see the seventh step. There 1,3-bisphosphoglyceric acid undergoes dephosphorylation. In this step, one ADP converts into ATP. In this step, ATP is going to produce. Okay, let's see the eighth step. In this step, 3-PGA undergoes intramolecular shift. Okay, so phosphate group is shifted from third carbon to the second carbon. That's why 3-PGA converts into 2-PGA in presence of an enzyme mutase. So this step is called as intramolecular shift. So let's see the ninth step. There 2-PGA in presence of an enzyme enolase. So it undergoes dehydration process. It undergoes dehydration, it loses water molecule, then 2-PGA can converts into PEP, phosphoenol pyruvic acid. So now let's see the last step. There PEP undergoes dephosphorylation again in presence of an enzyme pyruvic kinase. Here also one ADP converts into ATP. So in this step also ATP is going to synthesize. This step is called as dephosphorylation. Here PEP is converts into pyruvic acid. So from 6th step to 10th step, these steps occurs 2 times. So that's why we are getting every molecule 2, 2, 2. Okay, you have to multiply each molecule into 2. Okay, so that's why if you see the energetics of glycolysis, so in the first and third steps, 2 ATPs are utilized, whereas in the 7th and 10th steps, 2 ATPs are produced. These reactions occurs 2 times. 2 into 2 is equal to 4 ATPs are produced. So we have to remove these 2 ATPs from the 4 ATPs, then we get net 2 ATPs are produced from the glycolysis directly. Not only that, we also get 2 NADPHs. Okay, so there are also energy molecules. So let's see the salient features of glycolysis. So it occurs in the cytosol. Here one glucose molecule is undergoes partial oxidation, can converts into 2 pyruvic acid molecules. Okay, so no oxygen is used in this process and no CO2 is released in this process. Here it occurs, here glucose undergoes partial oxidation only. So actually glycolysis is common for both aerobic organisms and anaerobic organisms. Okay.